I've really always tried to have a vector to it, something that if you do it, it could really help answer a fundamental question to give people tools to have a better understanding of how things work. I started my lab on protein engineering, and that was the chemistry side of things because I could synthesize oligonucleotides, and there are only a few people that could do that. And I applied it to proteins with known structures. So that was site-directed mutagenesis. That area has now gone into hyperspace 40 years later with the ability to do machine learning and the computational approaches that are allowing protein engineering to really live up to its original goals. But I couldn't wait for that. So the structure-function relationships of understanding how enzymes work evolved in my lab to structure-based drug design. So I led a team to help determine the structure of HIV protease, one of the early structures, and then use that structure to identify small molecules that could bind to the active site. So that was the beginning of structure-based drug design. I'm really proud to say that I contributed to an enormous field of the current drugs that are in the clinic right now that target HIV protease and are part of cocktails that have really brought what used to be a death sentence when I was starting my lab into something that's a manageable disease. So I was a brick in the wall, maybe I was an arch, maybe I was a doorway. That was very fundamental work that had some contribution to the bigger picture of things. More recently, we've developed some tools that are imaging agents that allow you to follow proteolytic activity in vivo. Now, why might that be interesting? Turns out it's a sensor that's now being used in the clinic to monitor whether T cells are active or not. So checkpoint inhibitors and approaches that take the brakes off of the immune system are breakthrough drugs, but they also have some problems of a lot of side effects. So knowing when the proteases are active from the T cells allows a clinician to decide whether to continue the treatment or to back off and not use it if it's not being effective. So I'm very excited about that technology because it can be used not only for imaging, but delivering therapeutic payloads, the pro-drug idea. There are a number of companies that are doing that type of work of using proteolytic activity to activate a quite toxic drug, but mask it and increase its therapeutic window. This one that we're working on now is to go after intracellular targets like tumor-specific antigens, in particular KRAS, which is in 20% of cancers. But it's an intracellular target, and there's breakthrough drugs that have been developed that covalently target KRAS and form a covalent bond to inactivate its activity. What we've done is we've shown that irreversibly bound molecule attached to KRAS gets degraded by the proteasome and presented onto the MHC class 1 antigens to give you now a chemically modified neoepitope that can be recognized now by immunotherapy. So we're taking intracellular targets and approaching them with immunotherapy. So these last two projects have helped me learn about immunology, and it's a fascinating field that I'm learning through building tools to help bring teams together to go after these sorts of things. So if this really works, it could lead to eventually potential cures, because the sotorasib things that Kayvon Chokat's been developing that go after these intracellular targets are breakthrough drugs, but resistance comes up very quickly. So this is a way to overcome resistance. What we do, we try to do well, but sometimes it even answers a question to help move the balls a little bit more forward. All of these projects that I'm talking about are collaborative. The way I've been able to contribute is I do what I do well and work with fantastic people, and we get 1 + 1 to be more than 2 sometimes. Synergy, I think, is the right word.